Hello.
Hello, Mr. Paulo.
Yes, very well. Sound and the image.
Yeah.
Okay.
Everything looks perfect.
Okay. Your nanomedicine is on the screen.
Yeah, nanomedicine.
All right.
Yes.
Good.
Very well.
So this is the
So
total. When doing introduction, use this image. After introduction, you need to click the PowerPoint.
Okay.
And
yes,
start input.
Yes.
I will start the program now. One moment.
For your listeners, after this, you will go into the slide.
Kenapa tak nampak muka dia ke sis? Why can I...
Bom dia, boa tarde, boa noite aos internautas que nos acompanham neste programa Nanoalerta, hoje no seu número 205. Fazemos este programa dedicado ao público não especialista, porque é este público que paga as pesquisas sobre nanotecnologia realizadas no Brasil. Também fazemos este programa porque é aqui que nós podemos discutir os impactos da nanotecnologia na sociedade, no meio ambiente, na economia, na saúde, coisa que não são discutidas aonde se faz nanotecnologia no Brasil. Por fim, fazemos esse programa porque acreditamos que ser livre é ser bem informado e a divulgação científica contribui, colabora para o aprofundamento da democracia e da cidadania no Brasil. Conforme tínhamos anunciado, estamos conectados com a doutora Latifah Saiful Yazan, da Faculdade de Medicina e Saúde, diretamente de KUALALAMPUR, na Malásia. Então, eu vou passar a palavra para ela, ela vai se apresentar primeiro e depois começar a apresentação dela. Ok, Latifah, could you please make your self-presentation about your experience with nanotechnology?
Thank you, Mr. Paulo. Hi, everyone. I'm Latifah. I am a lecturer and a researcher from University Putra, Malaysia. So for your kind information, University Putra, Malaysia is one of the research universities in Malaysia. So it means we focus so much on research. So basically my area of expertise is molecular and cell biology or medical biotechnology. And my research interest is drug discovery, especially from natural products. It is just very recently that I have started research in nanomedicine, simply because we have a collaboration with a friend of mine who has actually developed a nano delivery system for other drugs. Now we have a very potent compound known as pymokinone. So we want to see whether or not with the help of a drug delivery system will enhance the therapeutic ability of our compound of interest. Thank you.
Okay, now you can start your presentation.
Okay, we'll do it. Okay, so the topic of my presentation is nanomedicine. Again, I'm Latifah Saiful Yazan from Department of Biomedical Science, Faculty of Medicine and Health Sciences, University Putra, Malaysia. This is actually a brief explanation or a brief definition of what nanomedicine is. It is actually the medical application of nanotechnology. As we all know, when it comes to drug administration, oral administration is the most preferred one, simply because it is the most convenient, it is non-invasive, and it is of the lowest cost. But unfortunately, when it comes to oral administration, so it has several challenges, especially those of protein-based drugs, because they are very sensitive to acid and enzymes that are occurring in our gut. So due to that problem, when we have the drug being administered orally, it will be digested by the enzymes along the gut, and it will lead to low bioavailability. It means that it is not being efficiently used by the cells to get the therapeutic effects. And also, when it comes to oral drug administration, majority of the drugs available in the market nowadays are very lipophilic. It means that they are having a very low solubility in water, and this will also lead to poor oral bioavailability. So let me take an example of Alzheimer's disease. The main challenge of delivering drugs for Alzheimer's disease is that it has to cross the blood-brain barrier, or popularly known as the triple B. So one of the ways to tackle or to get through this blood-brain barrier is by the use of polymers, or in general you can use a drug delivery system to get your drugs into the affected area. So it has been coined by many researchers that nanotechnology is going to be having a major impact to the survival of the human race, simply because it can probably tackle a lot of diseases by enhancing the therapeutic ability of those drugs. So if I can summarize the benefits of nano delivery system, one, it is saving the use of additional drug, because with the use of a nano delivery system, you can actually target the area of interest, and you need to deliver the correct amount of drug. So in that case, you don't have to give the patients the dose that is not needed to cure the disease. So it means that in a way, lower dose is required. And with nano delivery system, we can actually control the release. It means that by controlling the release, you can increase the retention time, giving the affected area time to react with the drugs to give the full maximum effect. It is possible to do targeting, and then it is very cost effective, simply because when you can save the drug, it means that you have a lower cost of medication. And of course, when it comes to drugs which are probably very sensitive to acid and enzymes, as I mentioned earlier, so by having this delivery system, it can actually protect the drugs from being digested by the enzymes that will actually increase the bioavailability. So there are various nanoparticles available in the market. If I can summarize it in this figure, so it is actually classified into organic and inorganic nanoparticles. So some examples of organic nanoparticles are polymeric nanosphere. So liposome is very widely used in cosmetics. And for the inorganic nanoparticles, so we have gold nanoparticle, and it is also commonly used in cosmetic purposes. And very recently, people are very much interested in this quantum dot because it has a dual function. One is for diagnosis, and also it can help to deliver the drug of interest. So this is another figure of various so-called nanoparticles available in the market. So we have more here. Liposome again, solid lipid nanoparticle. Okay, and then we have a polymer nanocapsule and chitosan. So chitosan is being famously used now because this is a so-called cheap material. So you can get it from crustacean, from prawns. So they are using chitosan a lot in drug delivery system. So this is a very good paper if anyone is interested to read more about all these nanoparticles which are commonly used in the market nowadays. So in general, nanocarrier can also the organic nanocarrier can be divided into two. One is polymer-based nanocarrier. So these are the examples. Another one is lipid-based nanocarrier. So the examples are liposomes, the solid lipid nanoparticle, and nanostructured lipid carriers. So I want to inform that this is actually our main interest, the lipid-based nanocarrier. So this is a figure showing the evolution of the lipid nanostructured carrier. So here this is the solid lipid nanoparticle, so the first so-called generation. So as you can see here, solid lipid nanoparticle, it is made up of solid lipid. It means that if you can see here, so the structure is very perfect. So it is a brick wall structure. So it means that if it is too perfect, so there are no gaps that you can actually load the drugs in. So it means that with this system, it will lead to high drug expulsion during storage. By time, researchers are actually coming up with another generation of solid lipid nanoparticle called nanolipid carrier. Okay, so here theoretically this so-called carrier is a mixture of a solid lipid and liquid lipid. So when you have a combination of these types of lipid, so it creates imperfection in the structure that you can see some gaps are available in the formulation. So with these gaps, it means that you have more drugs to be incorporated. So this one will lead to very minimum drug expulsion and then also long-term drug stability. Let me bring your attention to our so-called material of interest, so which is a thymoquinone loaded nanostructured lipid carrier, and I will focus more on the anti-breast cancer properties. These are the people involved in this study. I really would like to acknowledge Rasidi Abdullah, Ng Wei Kiat, and Hao Chi Woon, who have actually developed the so-called delivery system, which is a nanostructured lipid carrier. So this is our compound of interest. It is a bioactive from seeds called Nigella sativa. This is a very potent molecule. It has various pharmacological properties, and in our previous study, thymoquinone has been shown to exhibit remarkable anti-cancer properties in vitro and in vivo. There's one issue of thymoquinone is that it has to be administered via intraperitoneal route, which is having some challenges when it comes to preclinical and clinical use due to high discomfort, costly and sterility issues. Well, oral delivery of TQ is possible, but it is very limited by its solubility related poor oral bioavailability. It means that thymoquinone is very lipophilic. When it is lipophilic, it is having low solubility in water, which is not possible for the cells to take up, and then this will lead to low bioavailability. So we have actually developed this structure called thymoquinone lipid nanostructured lipid particle. So we have filed a patent for this. So this is the figure. So you can see here the drug is being encapsulated in the middle of the structure. So it is the use of a combination of liquid lipid and solid lipid. So this is how we synthesize TQ-NLC. So we have two matrices. One is lipid matrix. So these are the components of lipid matrix. We have the solid lipid, which is hydrogenated palm oil or HPO. So this is the byproduct of our oil palm industry. And we have liquid lipid, the famous olive oil. We have lecithin, and we have our thymoquinone here. For the aqueous matrix, we have sorbitol that will give the sweet taste to the formulation. We have polysorbate 80, the surfactant. So we have thimerosal that will act as as a preservative agent, and we have water. So these two matrices are being mixed up, dispersed, and subjected to high-speed stirring, then high-pressure homogenization, and then recrystallization. After we have synthesized TQ-NLC, so we have to do all these physical-chemical characteristics that include the average diameter, the polydispersity index, the surface or zeta potential, and then drug encapsulation efficiency and loading capacity, and also we need to check the stability at room temperature. Then after characterization, we subjected the nanoparticle to all these tests to see whether or not they will show enhanced anti-cancer properties. So the assays involve in vitro analysis where we tested it out to a few cancer cell lines. Here are cervical cancer cell lines, and also the breast cancer cell lines. And we also studied the drug release using this technique, and then we see whether or not we can actually protect the drug from being digested by using this gastric digestion assay. We also did acute and subacute toxicity as to ensure that our TQ-NLC is not toxic. And finally, we determined the anti-breast cancer properties. So this is how it looks like. This is the carrier, the nanolipid carrier, and this is how it looks like. It is yellow when we have actually encapsulated thymoquinone in NLC. Thymoquinone is actually yellow in color. Then when we viewed under a transmission electron microscope, you can see that our so-called formation is quite homogeneous. We can see majority of TQ-NLC molecules have the diameter less than 50 nanometer. So when it comes to average diameter, it is around 36 nanometer. So it has been reported in many literatures that a particle with a diameter less than 100 nanometer will exhibit unique physical and biological properties. So this will also minimize the probability to be phagocytosed by macrophages. It means that it is not being cleared out from our system. So when you have a molecule which is small, it means that it has a larger surface area that will lead to maximum reaction with its target component. So it means that we are expecting that the biological properties of thymoquinone will be fully utilized when it has been encapsulated in NLC. So the polydispersity index is around 0.17. So it means that with this value, the nanostructured particles are in monodispersity and homogeneous with narrow size distribution. So it means that there is no aggregation of the nanoparticles. So one aggregation is existing, it means that you have a bigger molecule. So this will actually impede the targeting efficiency, and then aggregation will also reduce cellular uptake and cytotoxicity because an increase in the particle size, which will decrease the surface area. So once it is being aggregated, it means that the whole thing will be settled out of suspension and it is no longer bioavailable. So the zeta potential, it is not ideal. If you can see here, it is minus 16.72 millivolt. So again, it is not considered as a good value because the ideal one would be more than 30 millivolt or less than minus 30 millivolt. And in a way, with this value, our formation is considered not stable. But with the use of polysorbate 80, so in a way it helps to stabilize our nano formulation. And it is of advantage because polysorbate 80 is of low toxicity according to US FDA, which is given the status of GRAS, generally regarded as safe. So when it comes to drug encapsulation efficiency, so it shows a very remarkable drug encapsulation efficiency, I would say, because we managed to encapsulate 90%, more than 97% of thymoquinone in NLC. Again, the reason could be the use of a combination of lipid, which is HPO and olive oil just now, that it will create a very good core to accommodate this thymoquinone, which is lipophilic. And the imperfection in the so-called structure lowers the probability of TQ to be exposed upon storage. So it means that with this high encapsulation and loading capacity, will avoid the wastage of the compound and therefore lowering the cost of production. This figure shows the cytotoxicity of TQ-NLC towards all these cell lines I mentioned earlier. So these two are breast cancer cell lines, these two are cervical cancer cell lines, and we also included the normal cell lines here as a reference. So we are based on the IC50 value to see whether or not tamoxifen or tamoxifen and TQNRC will be having good anti-cancer properties in vitro. So as you can see here, the IC50 value towards cancerous cell lines are lower to the cancer cell lines compared to the normal cell line. It means that it shows some specificity in activity. The lower the IC50 value, the more toxic the material is. So it means here, so it is very toxic to the cancer cell lines, but it is less toxic to the normal cell lines. This figure shows the cells treated with TQNLC. This is the one untreated, and this is the one treated with TQNLC. So the cells are showing characteristic of apoptosis. So you can see here it is cellular shrinkage. So we can see membrane blabbing. And then when we did the cell cycle analysis, the TQNLC is, TQNLC arrested the cells at G2M and S phase. It is very important to note that whenever we encapsulate a material in a delivery system, it shouldn't change the activity. So it means here, tamoxifen alone will arrest the cells at G2M and S phase. And when we encapsulated tamoxifen in LC, it shows the same property. It arrested the cells at the same phase. Okay, and it is of advantage when a material is inducing apoptosis, because we know apoptosis is a preferred mode of cell death because it doesn't trigger inflammatory response to the neighboring cell, which is different from necrosis. We also did the release kinetic of TQ from NLC. So it shows that the release kinetic follows the zero order. So with this zero order release kinetic, so it shows that the formulation is an ideal delivery system where it allows a controlled drug release. So with controlled drug release, it means that we can maintain the drug concentration within the therapeutic window. And in that case, we can actually minimize the episode of toxicity of tamoxifen. So we also did some assay to see whether or not TQNLC will be digested in the gut and in the stomach. So this simulated gastric digestion, here we can see that tamoxifen, TQNLC is not yet digested. So this is what we have in our gut, and it is released when it goes to the intestine. So it means that from this in vitro analysis, NLC is giving a protection to TQ from the pepsin digestion and degradation. So it means that when it is passing through the low pH, it is being protected, and it is only being released in the stomach, all right, and until it reaches the small intestine. So with what we have achieved, with the data that we have achieved from these two assays, we believe that when it comes to the real system, which is human, so in the small intestine, TQ will be released from NLC, and then later it will be solubilized in mixed meat cells, which consist these two products. One is bile salts, another one is lipid degradation products, such as fatty acids and glycerol. Okay, so this will actually enable the large amount of TQ to be solubilized in the medium of small intestine, and all this actually will result in high bioavailability and bioaccessibility. It means that it will be used very efficiently by the cells, and then the person will experience a so-called therapeutic maximum therapeutic effects. So we have done the toxicity of TQ-NLC. So it is obvious here. By encapsulating TQ in NLC, so at first TQ falls in category 3 with the LD50 between 50 and 300 milligram per kg. But when we encapsulated TQ in NLC, it falls in category 4, which is LD50 of more than 300 milligram per kg but less than 2000 mg per kg. It means that TQ-NLC was less toxic compared to its original form, which is TQ. So we can actually conclude from here that encapsulation of TQ with lipid carrier reduces its toxicity. So this is a figure showing the tumor volume of animal with breast cancer treated with TQ-NLC. So from this figure, we conclude that there was a reduction of tumor volume by both TQ and TQ-NLC, and the reduction is comparable to the one of doxorubicin, which is the commercial drug used for treatment of breast cancer. So this figure shows metastases to the lungs. Okay, so the red dots are all metastatic cells. So we can see here, so this is the normal one without cancer and untreated. So this is the negative control where the animals are having cancer but not treated with any material. This is the positive control. So this is treated with doxorubicin. So we have here TQ-NLC at 25 mg per kg. This is thermoquinone. This is at 50 mg per kg, and this is 100 mg per kg. So we can see that inhibition of lung metastasis by TQ and TQ-NLC at 25 and 50 milligram per kg is comparable to the one of doxorubicin. So we can see less number of tumor nodules existing in the lungs. And also we can see that TQ-NLC at 100 mg per kg really improve the survival of the mice having breast cancer. In conclusion, from our in vitro and in vivo analysis, we believe that the use of NLC enhances the anti-cancer properties of thermoquinone, and at the same time reducing its toxicity. Thank you.
Okay, Latifah, thank you so much for your presentation.
Thank you.
Was very complete and so good. I have some question for you. Not a specific question because I don't have in-depth knowledge to make some in-depth question. I have many general question.
Okay, sure.
Okay, the first general question is about the toxicology.
Toxicology.
Yes, and the nanotoxicology.
Nanotoxicology.
Yes. My question is if, in your view, the theoretical framework of the nanotoxicology is the same the theoretical framework of the toxicology.
If I understand your question correctly, so you mean whether or not a nano formulation will be having the same toxic effects when it is if we were to compare nano formulation with its original compound, will it give the same toxicity? Is that your question?
Yeah.
Okay. It is quite difficult to conclude whether or not nano formulation will give toxic effects the same as what the original compound will give. And because, as we know, because nanoformation, it is of a small size, and it can actually evade an immune system. So it means that it is not going to be phagocytosed by the immune relevant molecules. There is a high possibility that it will actually accumulate in the liver. Probably we cannot see the effect in short term, but in long term, probably it will damage the liver more badly compared to the compound which is not being encapsulated in any delivery system.
Okay, and the second question is about the bottleneck in this kind of research. What is, in your opinion, the bottleneck in this kind of research? The big challenge to develop more this kind of research?
The big challenge, I would say, is to determine which delivery system is suitable for the compound of interest. Let me give you an example. When it comes to NLC, we think that all lipophilic materials will go into the delivery system. But it is not the case, because last time we have tried another compound which is very lipophilic, but unfortunately it doesn't go into the delivery system.
Okay.
Because we have we have many systems that we have to try it one by one. Even we have tried to actually tag our thymoquinone with a carbon nanotube, it didn't work, unfortunately. I would say that is the biggest challenge in doing nanomedicine.
Okay, and how much research is necessary to do this kind of the research? I don't know, in your team, how much person is in your team? My question is about your team of research has 5, 10, 20 persons. How much?
How many you mean?
Yeah.
At the moment we have 5 people. But if I were to do more, then I need more people to be in my group. But to my thinking, who you need in the group is one who can actually develop the delivery system. I think we need a chemist in that case. Of course, when it comes to animal work, you need to have a pathologist. And then when it comes to human research, then you need a physician probably. But what we have now, I think, would be sufficient at the moment to study TQNLC in preclinical setup.
Okay, and the budget to do this kind of research is a very expensive research or not?
It is very expensive, Mr. Paulo, because thymoquinone is already expensive. Now you want to have it in a delivery system, that is another expensive thing. And usually biomedical relevant researches are all expensive, because we have to buy the cell lines. So the cell lines are not cheap, that you have to maintain them in very special medium, very special environment. And when it comes to animals are also expensive, and they have to have a special care. So it is not yet into human research. So human research, I would say, is more challenging, that you need a lot more research grant to support.
Is possible to substitute the animals for computer simulation?
At the moment, I will say no, but I know that some researchers are developing this so-called organ on chip. But to my thinking, it is still not representing the whole animal. Even cell lines, you cannot use the cell lines to and extrapolate the data to be used in a so-called animal setup. Because in animal or in human, so we have many organs, and the two important organs which are actually um, influencing the activity of the drugs are liver and kidney. So if you were to use cell lines, you cannot have these two organs. So if you have the organ on chip, also you cannot have a complete system. So that's why in many cases, you may get a very contradicting data whereby probably you do in cell lines, it shows very good activity, but when you go into animal model, it doesn't show any activity, and vice versa. So that is the so-called understanding we need to have in mind before we come up with any drug for treatment of diseases.
Okay, and you think that will be possible in the near future we have nano robots in our blood to fix any organs of human beings?
I think some researchers are working on that. Even in Japan, so they have all this AI, so-called artificial intelligence technology, where they have come up with robots. I think it is possible. People are getting cleverer by time. So they have actually invented a lot of so-called very good invention. So I think yes, one day we'll have all these so-called robots, and probably the concern is that probably it will take up the role of human, and we're not doing anything. So we let the robots to do everything for us.
And about the money, your budget, is a public money or is a private money?
Again, pardon me?
Yes. My question is if in your research you have public money or you have private money?
Majority of the research grants come from our government, but we are also applying for private grants. So I have both.
Both. Okay.
Yes.
So you think that you have enough budget to do this kind of research?
It is not enough. Well, because research is never-ending journey, I would say. So the moment you reach to certain stage, then you have to continue and move on. For example, in our case, we started off with cell lines. So we see that the nano formation is showing good activity in cells. Then we move on to animal model. And of course, it is a dream of us just to have their research material to be translated. So hopefully one day, who knows, we can actually test our T-pregnancy in human and see how the effects are.
Okay, I can tell you that your interview was so good. I put my question and I have your answer. So was a very good program, and I hope you continue to work with this kind of research. And when you have have news about your research, he can do another interview.
Yeah, yes. And thank you so much for the opportunity to be connected to you.
Yeah.
So it is really a good platform for us to share what we have been doing so far. And I hope with this we can have probably collaborations from your side.
Yeah.
From Brazil, then we can see how much we can do to improve the quality of our research.
Very good. You are my first interview from Malaysia.
Really?
Yeah.
Wow, thank you so much. Thank you so much. After so many delays, last time we were supposed to be on air, but there was a miscommunication when it comes to time. So it is at night in Malaysia, and you are now at daytime.
Okay, you can suggest me another research from Malaysia that is working with nanomedicine or with nanotechnology. I would like inviting your research suggestions.
Yeah, sure. I have a friend who is doing a lot on nanotechnology. So she used a quantum dot. So as I mentioned earlier, quantum dot is very special because it can have dual function. It can go for diagnosis and also for drug delivery. I will email to you her details and then let you communicate with her.
Okay, thank you so much for your contribution to our program.
Thank you so much, Mr. Paulo, again for the invitation and the opportunity.
Okay, bye-bye.
Bye, see you.